Okay, let's start. Okay, today we invited Dr. David Alvarez-Ponce from Department of Biology. He is the best one in this university on the west side about the evolution and genome and genes. And he's doing a great job. And then a lot of publications came out or regarding the evolution. And then today he will give us so he will share some expertise and researches about his work. And please welcome Dr. David Alvarez-Ponce. And please go
Thank you,
ahead.
everyone. Thank you, everyone, for such a nice presentation, better than I deserve. So I'm going to share my screen.
Yeah.
The title of my presentation is Evolution of Genes and Gene Networks, but more specifically, I'm going to try to explain different aspects of how the position of genes within networks affect their evolutionary patterns, the way in which they evolve. And what do I mean by that? Just to give you a first snapshot of what this talk is about. Say this is a network, okay, or this is a network, for instance. So one of the things that we've been learning throughout the years is that genes at different parts of this network, for instance, those that are highly connected or those that are upstream of signaling networks, they evolve different from others. Okay, the position of genes within a network make them evolve faster or slower or duplicate more or duplicate less. And this is what this talk is going to focus about. So first of all, I'm going to give you an introduction to networks. Then I'm going to talk about how genes at different positions of these networks evolve at different rates. Some evolve fast, some evolve slow. Then I'm going to talk about positive selection, how some genes within the network and certain positions are more likely than others to be under positive selection. And also, if we have time, which I don't know if we will, because we have only 50 minutes, about duplicability, why some genes duplicate and some genes don't, and how the network position of these genes affect their duplicability. So first of all, what do I mean by a network? Here, this is the insulin pathway in Drosophila. This is the insulin receptor. These are the Drosophila insulin-like peptides. And you see how all of these proteins, they interact with each other, right? So the insulins activate the insulin receptor. The insulin receptor, once activated, it recruits another protein that then recruits another complex of proteins and so on. And there's a cascade of events, okay? So this is what I mean by a network. In this case, a signal transduction network. Or here we have different pathways, biosynthetic pathways that you are familiar with. In this case, there's not necessarily a physical contact between the different proteins in this pathway, but what they do is to catalyze reactions from one from one substance to another, and then the product of that reaction is the substrate for the following reaction. Okay, so that's how the different enzymes here or here or here are connected to each other. This is part of the human signal transduction network. In this case, you see how for many of these proteins, there are lines that connect them. That means they physically interact. And on top of that, this data set is particularly rich because some of the interactions, they are represented by arrows, which means that this protein activates this other protein or it inhibits this other protein. So this is a network that we will see what that means. It's a directed network. Some of the interactions in it, they have a direction, okay? We have protein-protein interaction networks. So here I'm moving from a small scale to large scale. And this was the protein interaction network for yeast quite a few years ago. Now we know much more interactions, but if I show them all, it's hard to even see a network, right? So I prefer to show you a slightly older version of it, because in this one, we can see how, for instance, some proteins like this one are very highly connected, or others like this one here are much less connected, okay? And this is the metabolic network, which is the addition of all of the metabolic pathways, like the biosynthetic pathways that I've been showing you a moment ago. So this is what we're talking by networks. Also, we have gene regulatory networks in which we have transcription factors that activate other transcription factors and other genes, and they can be arranged into a hierarchical structure. Okay, so to study networks, we use graph theory or network theory, which is a branch of graph theory, is a branch of mathematics that deals with the structures like this. In a graph, we have nodes that, like this one or this one, that represent entities. In our case, they represent proteins, but graph theory could be used to represent cities and their connections, or people and their friendships, or any other thing. And we have edges that represent relationships between them. In our case, often these relationships, for instance, in the case of protein-protein interaction networks, they are going to represent physical interactions between the proteins. Here, if 4 and 6 are connected, that is because they physically interact at some point. Even though not all of the networks that we're going to see, not in all of them, edges mean the same. Okay, so you have to, when you look at a network, you first have to ask yourself, what do nodes mean in this network, and what do edges mean in this network? In this network, this is the general representation, the most basic representation of a network. That's what we call an undirected graph. We can distinguish, we can calculate for each of the nodes what we call degree, which is a measure of centrality, which is the number of interactions that each of the nodes has, or the number of edges that each of the nodes has. For instance, in this case, for node 6, degree is 1 because it only interacts with node 4, or for node 1, it is 2 because it has 2 edges. We might be interested in some circumstances to model graphs in a slightly more complex way, which is directed graphs. We can use directed graphs in which we don't talk about edges, we more generally talk about arcs, because they are directed interactions or relationships, and we represent them with an arrow. And in this case, that could mean that node 6 activates node 3, or imagine that could mean that from city 6 you can go from city 3 to city 3, but not the other way around, and things like that. And in this case, we can also calculate degree, but we can also calculate in degree and out degree. The in degree of a node is the number of incoming arrows. Say for, in this case, for 6, the in degree would be 1, and the out degree is the number of outgoing arrows. For instance, for 2, it would be 3. Okay, out degree would be 3. And these metrics, degree, in degree, out degree, they give us an idea of the hierarchical position of a node, whether within a network, or how important a node is within a network, okay? How it can influence the rest of the network, very generally. Okay, so we're gonna be talking a lot about protein-protein interaction networks, and one source from which you can obtain this network data is from the BioGrid database. For instance, and it has information for many, for a bunch of species, I would say. For most species, we don't have a lot of protein-protein interaction data, but for the model organisms and for human, we have quite a bit. And this is from a few years ago. This is already old data. This is from 2012, actually, so pretty old data. And this changes every month. If you look at these numbers, they are gonna be different. So I should have updated this slide, but never mind. In 2012, we had for Arabidopsis thaliana, we had over 16,000 protein-protein interactions, physical interactions, 188 interactions of another kind, and here you have a bit more information. And this is how the network, the Arabidopsis protein-protein interaction network looked back at the time. This is a network that I reconstructed from the data at the BioGrid. And this is the network for a human. Do we have a question? Sorry, I thought someone was going to ask a question. So this was in 2011, the human protein-protein interaction network, which I also obtained from the BioGrid to do different analysis. Another source of network data is the Kyoto Encyclopedia of Genes and Genomes database. And in this database, this is different because it's been more manually curated, and we have charts like this one. This belongs to the insulin pathway. And here we have the different interactions. You see how many of them are represented by arrows or by this other symbol, which means inhibition. And there's a team of people working to put these charts together and to curate it. Some of this data comes from, most of it comes from the literature, but as well other data has been inferred from other species. For instance, if you look at the insulin signaling pathway for a species that it's not very well studied, like I'm going to say as an example, a frog, for instance, or a pica, for instance, is going to be, for the most part, the information is going to be derived from species that have been well studied, like human. Okay, so this is another source, another database in which we can find network data. So where does this data come from ultimately? So at some point, someone is generating this data in the lab. And by the way, these networks are collective efforts from hundreds of scientists, each of them describing a number of interactions. They are the result of a small scale analysis. That means a lab focusing on the interaction between one protein and another protein, establishing if they interact or not and how, and also large scale analysis in which we're talking about screening hundreds or thousands of interactions at a time. And they use different techniques. Here we have different examples, and I'm going to talk a little bit more about the yeast 2 hybrids technique. This technique is used to study whether or not two proteins interact. In this example, the proteins are called X and Y, okay? So you want to know if X and Y interact, or here also they are called bait and prey. It means the same, okay? So what you do is you create these plasmids, and you have two of them, and in one of them you clone, you incorporate the coding part for that encodes for of the gene that encodes for protein X, and you put it right next to another coding element that encodes for part of a transcription factor. And then you do the same with the other protein. You put here the gene encoding protein Y, and next to it and in frame, you put part of a gene encoding for a transcription factor, and that part encodes for another part of the transcription factor. So when you put these two plasmids into a yeast cell, the yeast is gonna express these two proteins, one which is your X protein next to a domain of a transcription factor, and this is the binding domain, and the other next to the activation domain of the same transcription factor. Now, if the two proteins interact, if they happen to interact, which is what we have here, prey and bait interacting to each other, then because prey is going to be bind to the activation domain and the bait is going to be bind to the DNA binding domain, then the whole system is going to act as a transcription factor. And if you have here a reporter gene, remember that's a gene that you can see easily if it's expressed or not. Maybe it confers antibiotic resistance or a certain color or fluorescence. Then if the reporter gene is being expressed, that means that the two proteins do interact. If they don't interact, then the whole system is not going to form and you are not going to see expression of the reporter gene. Okay, so that's one of the ways by which we can tell if two proteins interact or not. So a few words about the properties of networks very quickly. I've been talking to you about degree, so how many interactions each protein has. If you look at the degree for all of these proteins in the human or any other protein-protein interaction network, and you do a histogram like this, so how many proteins have one interaction, how many proteins have two, and so on, it's going to follow this distribution, what we call a power law distribution. You're going to have that most proteins are going to have only one interaction. Then much fewer are going to have two, and fewer are going to have three, and this has a specific response to a specific formula. Many networks, not only biological, but also engineered or made by human beings, they exhibit this power law distribution. And it's under discussion in the case of biological networks. Why do we observe that? Is it adaptive or not? Another characteristic is modularity. In biological networks and also in other networks, we often find modules. Modules are groups of proteins that interact a lot with each other in the same module, but they interact less with proteins in other modules. Here we have one module represented in green, another in red, and another in yellow. In this other example, there's less modularity because there's more crosstalk between the different modules, and this is a more kind of real life scenario in which you can distinguish the modules, but also some crosstalk between them. You can think, for instance, as an example of these modules as different pathways in a metabolic network or in a signal transduction network. Another characteristic is motifs. If you look at the network and you look at the number of motifs like that, this is little subnetworks that have this topology. So if you look at how many times you find this or this or this topology in this big network, you're going to find that some of them are overrepresented. They are found more often than you would expect in a random network. Okay, so that's another characteristic that it's under discussion. Why do networks have it? Okay, so now I'm going to talk about rates of evolution, what they are, how we measure them, and how they distribute across networks. So here we have an alignment of two proteins. One is the human protein and the other is the mouse ortholog. Human and mouse diverged around 75 million years ago. And you see how in this alignment spans two lines. This specific protein is identical between human and mouse. So in the last 75 billion years of evolution, there's been no evolution in the protein sequence of that protein. On the other hand, this other protein, in the same amount of time, because we're also comparing human and mouse orthologs, it's changed quite a bit. It's changed a lot, okay? And also you see some parts that change more than others. For instance, this has been conserved, whereas this has changed quite a bit. So why is that? Why do some proteins evolve faster than others? This is the main question that my research focuses about. So one of the reasons could be that they are located in areas of different mutation rates. The genes encoding these proteins might be, imagine that this one is located in a mutational hotspot and this is not, okay? So that could be a reason. That's not the whole reason, of course, but you have to discount that effect of the mutation rate. And how do we do it? We study separate synonymous sites and non-synonymous sites. What are synonymous sites and non-synonymous sites? This is 6 codons of a gene encoding for a protein, right? And this could be, the first one could be the sequence in human, and this other one could be the sequence in mouse, for instance. And then you see next to the codons, you have the amino acids that they encode. And I've marked in green the different substitutions, the positions at which one species has one nucleotide and the other has another one. And this one, human has a C and mouse has an A. So at some point of the evolution of that codon, there was at least one mutation that was fixed. But in both cases, both codons encode for the same amino acid. So that's what we call a synonymous substitution because it doesn't change the protein sequence. This is another example. And this is an example of a non-synonymous substitution because in this case, for this codon, changing an A by a T results in different amino acids. In one case, you have leucine and the other you have phenylalanine. So this is a mutation that does change the protein sequence. We can calculate the degree of divergence of non-synonymous sites. It's the N. This is the number of non-synonymous substitutions divided by the number of non-synonymous sites in the gene. And we can do the same for synonymous sites. And then we can calculate the ratio between both, which is what we call omega or DNDS ratio. Synonyms, they are synonyms. And this ratio can be equal to 1, and that's what we expect in a pseudogene. If there's no natural selection acting on the protein sequence, then natural selection is going to treat equally synonymous mutations and non-synonymous mutations, and they're going to fix at the same rate, okay? So then DN and DS are going to be equal or almost equal, and then omega is going to be 1, okay? So this is what we expect under no natural selection. For functional genes, for genes that are not pseudogenes, we most of the time are going to observe an omega ratio that's lower than 1. And that reflects negative selection or purifying selection. And that's because in these cases, DN is smaller than DS, and that's because non-synonymous mutations, those that do change the protein sequence, are filtered by, most of them are filtered by purifying selection or negative selection because they are deleterious. They have negative effects on fitness, okay? Whereas synonymous sites, they are often neutral. Sorry, synonymous substitutions, they are often neutral, typically changing one nucleotide by another. If that doesn't change the protein sequence, it's going to have no effect on fitness. So typically, synonymous sites are going to behave neutrally, whereas non-synonymous sites, mostly changes in them are going to be deleterious. So that's why we find this ratio. Sometimes we find a ratio higher than 1, and that's uncommon to find for entire genes. Sometimes it's specific parts of the gene that have this, and that's what we expect under positive selection. And I will talk about positive selection a little bit more later if we have time. So basically, in this study, this is from a paper in which they compared pairs of orthologs between Arabidopsis thaliana and Arabidopsis lirata. And they calculated Ka, which is to say Ka and Ks are the same as Dn and Ds. They are different ways to express the same quantities. And then this is the ratio, omega, okay? And you see how this ratio varies quite a bit. In this case, the mode is around 0.1, but you have proteins that have an omega of 0, like this one, that didn't change at all. And you have others that have an omega even above 1 or around and sometimes even above 1. So there's a lot of variability. Some proteins evolve very slow and some proteins evolve very fast. So why is that? One of the factors that we've learned over the years, in the last couple of decades, that it affects rates of protein evolution quite a bit is expression level. In this graph, we have expression, it represented as in this case, is the number of messenger RNA molecules per cell, and DN, in this case, they are not using DN-DS, but they are using DS, and they both correlate a lot. So basically, this is the protein rate of evolution. And there is a negative correlation, which means that highly expressed proteins, they tend to evolve slowly, okay? And it's quite a big question in my field to try to understand why does that happen. It's not obvious at all why does that happen. This is an experiment that my my postdoctoral advisor Mario Fares did in his lab, and each of these tries to be, it represents a petri dish. Sorry, I did this drawing, and it's not, I know it's not the best, but it is a petri dish. I realize now how I could have done it much better. But so basically, these are the E. coli colonies, and what they did in the lab is every day they would, so from this E. coli colony, they founded two parallel lines, A and B, and then every day someone in the lab, they would pick a colony and they would use it to create, to populate a fresh petri dish, okay? So this is an evolution experiment in the lab. This happened in line A for 260 generations and in line B for 250 generations. And then every certain amount of time, they would get a sample and they would freeze it. And then he liked saying that he was keeping a fossil record of the evolution of this line. So one of the things that we did was to sequence the original genome at the beginning of the experiment, and then the genomes at the end of each of the two parallel lines, and then also we sequence some in the middle. So basically, what we did, one of the things that we did with this data was to calculate the DNDS ratio by comparing genes at the beginning of the experiment and at the end, and comparing essential versus non-essential genes. Essential genes are those that if you knock them out, the cell doesn't live or it can't reproduce, okay? So they are essential. Whereas many genes are non-essential. If you knock them out, the cell can survive and reproduce, at least in the lab. And you would be surprised how many genes are actually non-essential. And there was a discussion, and there's still ongoing, on whether essentiality of genes impacts significantly or considerably rates of protein evolution. And in our study, we did find in one of the lines significant and quite strong differences in the rates of evolution of essential genes here in black and non-essential genes. So essential genes evolved slowly compared to non-essential genes. In the other line, we didn't find significant differences, maybe because they are not, or maybe because that line evolved more slowly than the other. So what happens with rates of protein evolution and networks? So here you have a protein that is very highly connected, and here you have another protein that is very lowly connected. Knowing nothing else about these two proteins, which of the two do you think would evolve faster? The highly connected one or the lowly connected one? No one? So the Mia? No?
Oh, I was thinking the lowly connected one would evolve faster.
That's what is expected, because the lowly connected one, this one, for instance, only interacts with another protein. So on the other hand, this interacts with many proteins, and this protein needs to, in order to function, needs to often to continue to interact with the other proteins. So if this protein here changes, it's very likely that it's gonna stop being able to interact with some of the other proteins. And if these interactions are important from the functional point of view, that's often going to be expected to be deleterious. Whereas this has only one other protein to interact with, so it's probably a smaller part of the fraction of its protein length that it's used to interacting with that protein, so it has much more wiggle room to evolve. Okay, that's part of it. So sorry. So we in the Arabidopsis thaliana protein-protein interaction network that I showed you earlier, we look at the correlation between omega and different metrics of centrality, and including degree, but I'm not showing you that in this slide. But in this case, we have betweenness. Betweenness is a slightly more complex measure of centrality. It's not as simple as degree. It has more to do with the global position of proteins within the network. But anyway, and we did find a negative correlation, but weak. Okay, it wasn't a very strong correlation. And in this other analysis, we classified proteins according to their betweenness, and then we calculated the median and other matrices of DNDS or omega in the different beings, and you see how the higher the betweenness, the smaller the rate of evolution, the slower they evolve, as Mia was predicting. Okay, but the correlation is pretty weak, and that's something that has been being discussed in the literature for a while. Some people think that the position of genes in the network has no effect or very weak effect, and that basically we observe these correlations due to other factors. Okay, so why is this correlation so weak? Do you have any suggestion? Well, it could be that actually the correlation is weak, or it could also be that this data, the protein-protein interaction network, it's very noisy. We know that the yeast to hybrids techniques and others used to collectively come up with this network, they are prone to errors, their biases, and so on. So many interactions have never been tested, for instance. Others have been tested a lot, so it's more likely that you're going to find them. You have false positives, false negatives, and so on. So that's one factor. Or maybe it's true that the correlation is very weak. So we did this study. This data is for human, and this is a principal component regression analysis. And we looked at the effect of different factors on the rates of protein evolution. And they are represented in different colors. And the ones in a black box here, so these are the different principal components, okay? And the ones that I marked with a black box, they are the ones that are related to position of proteins within the network, and see how out of the whole, so each of these lines all together, or bars, I should say, sorry, all together, they represent how much of the variability of omega is explained by the different factors, okay? And the fraction explained by a network centrality, it's not small compared to the other effects. Some scientists have defended the idea that it was almost only gene expression levels that explain rates of protein evolution, but we find that in this analysis, at least, network centrality has as much of an effect as position, or sorry, as expression, okay? Another thing that we did, we looked at, is to test the hypothesis that interacting proteins should have similar rates of evolution because of coevolution. So basically, if this protein here evolves fast, I would expect this other protein here to also evolve fast because they interact with each other, so they have to keep up with each other, okay? Very simplified. So what we did was to calculate for each interaction, we calculated the difference in the rates of evolution between the two proteins involved in the interaction, and then we divided that by the average rate of evolution of the two proteins, and then we average this metric for the whole network, okay? So if this network had, say, 14,000 interactions, so for each of the interactions, we calculated this difference in the rates of evolution, and we normalized it, and we did the average across the 14,000 interactions. And this is the value that we observed. The value was a little bit about 0.89. So is this value high or is it low? Is it within what we expect at random if proteins were randomly associated with each other, not related to their rate of evolution? So to test that, we compare that observed value with the values that we expect in a random network. So for that, we created, I don't remember if it was 1,000 or 10,000 random versions of this network with different constraints. And for each of these random networks in which proteins were more or less randomly distributed, we calculated the same quantity, and this is the distribution that we obtained. So the observed value is a bit below, and actually significantly below, because this surface here could be taken as being the p-value, below what you would expect at random. So there is a bit of that effect of interacting proteins showing similar rates of evolution, which possibly could be due to a coevolution. There's also other possible explanations that I'm not going to discuss here. Okay, so now I'm going to talk about signal transduction pathways and how the proteins within us, this is not a signal transduction pathway, about biosynthetic pathways and how proteins at different parts of these biosynthetic pathways evolve at different rates. So this is the anthocyanin pathway, and these are 6 enzymes that are key to it. And then in each step, you see, so you have this is converted into this, and this is converted into this, and so on. If you look at the rates of protein evolution, and this is something that these authors did in 1999 of these different enzymes, you see, and this is pathway positions, so positions 1, 2, 3, 4, 5, 6 are the ones here, and DN in this case is here. You see how there's a positive correlation in this case. So proteins acting upstream of this network, of this pathway, they tend to evolve slowly compared to proteins acting downstream of this pathway. Do you have any ideas why that could be? Why proteins acting here, they evolve slowly, maybe because they are more important for the pathway. So if you imagine you knock out this enzyme, then all of this is going to be affected. All of this is not going to be synthesized. Or if you mutate this enzyme, then potentially all of this is going to be altered, the synthesis of all of this. And imagine that all of that, which is, we believe it to be the case, all of these or most of these substances or compounds, they have an effect on fitness, okay? So mutating an upstream genes could be catastrophic, but if you mutate this downstream gene, then you're only affecting the synthesis of the final product. If you assume that each of them has for instance, an additive effect on fitness, then you expect a correlation like the one observed here. So this is for the anthocyanin pathway, but also in other pathways, similar trends have been observed. In these pathways, typically upstream genes are more constrained than downstream genes. So in my PhD thesis, I studied if the same was observed in signal transduction pathways, and I focused on the insulin pathway. In signal transduction pathways, you have a receptor, which can be in the membrane or not, and this receptor responds to signals, for instance, in the environment or from within the cell, and unleashes a response. As I told you, so I already talked to you a bit about the insulin pathway. In this case, the signal is the nutritional status, for instance, and then that triggers a cascade that ends up in affecting transcription and translation, among other things. And we studied the insulin pathway and the rates of evolution of their genes in 12 Drosophila species. These 12 genomes had just been published, and also in 6 vertebrates. I'm only going to talk to you about the 12 Drosophila genomes. Just very briefly know that similar or parallel results were obtained in the 6 vertebrates. So what we did was to look at the literature and come up with a list of genes involved in the pathway. And these genes are, they were in Drosophila melanogaster, which is by far the Drosophila species better studied. And this is the phylogeny of the 12 species. And then for each of these genes, we identified the orthologs in the different species, and more specifically, we focused on this group to calculate rates of protein evolution or omega ratios, and we focused on this group to avoid saturation of synonymous sites. Synonymous sites evolve fast, so we didn't want to compare species that were very different among them, because then it's possible that every synonymous site has undergone multiple substitutions. So we focus only in what it's called the melanogaster group for estimation of rates of protein evolution. And then from the network, or what we knew about the interactions between these proteins, we obtain this directed network. Okay, see how here interactions are represented by arrows, so it is a directed network. And we use that to encode, to come up with the position of its protein along the pathway, okay? So this is position 1, this is position 2, and so on, up to position 10, depending on how far away they are, far away, metaphorically, from the insulin receptor, far away in terms of information flow, okay? So we had this quantity that we call pathway position. And we found a correlation between pathway position here, represented from 1 to 10, and omega. But interestingly, it was negative. So it was opposite to the one that we had found, that other authors had found for biosynthetic pathways, okay? So we try to understand why we were finding a negative correlation, not a positive one. First of all, we looked separately at the evolution of non-synonymous sites here. So correlation between DN and position, and it was negative. And DS, in which case so synonymous sites, we didn't find a correlation. So that showed that it was protein evolution, not synonymous evolution, what was driving the correlation that we found. It had been said in the literature, showed to some extent that usually what they are called the regulatory genes of pathways, which are the ones not marked in red here, tend to evolve faster than what are called the effector genes, which are the ones encoding the proteins actually doing the job, okay, actually catalyzing the reactions and so on. So we said, okay, because the effector genes are downstream of the network, then, and if they evolve slow, then that could be driving the correlation that we observed. So we repeated our analysis removing these genes, and we still found a negative correlation, even stronger in that case. So that wasn't the reason. Very briefly, there are other factors known to affect rates of protein evolution. I have already talked about connectivity. Connectivity is the same as degree, by the way. I've also talked about expression level. I haven't talked about codon bias or protein length, but these are also factors affecting rates of evolution. So one of the analyses we did was this statistical analysis. This is called a path analysis, in which you represent the different causal relationships between the different variables. And then according to this model, so basically it's a way to trying to discount the effect of potentially confounding variables. So these are potentially confounding variables. But even accounting for these effects, we found a correlation, a negative correlation between pathway position and DN in this case. So we did, so this analysis suggests that it's not due to the distribution of connectivity across the network or expression level and so on. It seems to be a direct effect between position and DN. So as a conclusion, there is a negative correlation between position and rates of evolution in the insulin pathway, and a positive correlation in biosynthetic pathways, or at least the ones studied, generally among the ones studied at the time. So the next question was, is this general to signal transduction pathways, or is it something specific to the insulin pathway? Is it only the insulin pathway that has this different correlation? So another two studies after our study, this is the first paper we published, they also showed negative correlations in signal transduction pathways. That's the case of the Hawk pathway and also the Toll pathway. But still, 3 examples is not enough to establish a pattern. It could be just a random thing that the first 3 pathways to be studied showed a negative correlation. So I looked at a much bigger data set, which is, this is part of it, and I already talked to you about it, which is the human signal transduction network. And this is like 10% of it only, to say something, okay? So basically, in this network, I did different analysis. One of them was to look at proteins that they had a number of proteins downstream. Okay, so proteins that had an out degree higher than 0, okay? And then I calculated the omega for the upstream ones and the omega for the downstream ones. And I found, and then I calculated the median for the downstream ones. And then I found 800 cases of proteins that had at least one protein downstream of it, directly downstream of it, so directly activated or inhibiting it. And out of these 800 cases, in 446 cases, the rate of evolution was higher for the upstream protein than for the downstream proteins. And in 352, it was the opposite. And that is, we applied a binomial test, and that's significantly different. So there's statistically an excess of these kinds of situations. So that tells you that more often than not, the upstream protein evolves faster than its immediate downstream proteins, okay? So, which is, it's the same kind of effect, similar to what we found in the insulin pathway. So upstream proteins evolving faster than downstream ones. Another analysis was to compare the proteins that they were what we called extremely upstream. That means they had an in degree of 0 and an out degree higher than 0, like this one. It's not regulated by any other protein according to the data set, but it does regulate other proteins, versus the ones here represented in green, which we called extremely downstream. They are regulated by other proteins, but they don't regulate other proteins. So they have an in degree higher than 1 and an out degree equal to 1. And in comparing both, I also found significant differences. The upstream proteins on average, or the extremely upstream proteins like this one on average, they evolve faster than the extremely downstream. Another analysis was one in which I calculated for each protein how many proteins in total accumulatively lay downstream of it. So in this case, in this example, if you count how many proteins, I think it's 9, okay, that lay directly or indirectly downstream of this protein. So the activity of all of these proteins are potentially influenced by the activity of this other protein. Whereas for this other protein, for instance, it's only one protein that lays downstream of it, okay? So we call this amount H for hierarchical position or hierarchy. And this H showed a significant correlation with H, showing that proteins that are more highly hierarchical in the network, they evolve faster than proteins that are less hierarchical to the network. So all of these three analyses and others that we did, they showed that in general, in the human signal transduction network, proteins that lay upstream of it, they evolve faster than proteins that lay downstream of it. Okay, so same as with the insulin pathway. So what we had found in the insulin pathway was something to some extent more general to signal transduction pathways. So signal transduction pathways, in that aspect of their evolution, they behave different from biosynthetic pathways, which is basically that this is to show that this trend is not only specific to these three pathways, it's something more general, and it's opposite to the biosynthetic pathways. Okay, I don't know if we have time for more.
Yeah, David, we need to finish.
Okay,
Yeah.
so do you have questions? I made extensive use of Perl.
Yeah.
I had I had to, yeah, I programmed quite a bit to do this analysis. Then a little bit of R for statistical analysis. For some analysis, I've used also Python because it has a library called Network X, which you use for network analysis. So I think for things like betweenness calculations and things like that, I use that, but mostly Perl. Okay, cool.
So, any other questions?
Yeah.
No, no questions. I don't. Nobody has a question. So,
Okay.
David is like the older network you show it in the to do evolutionary analysis, and that is it, or it looks like a older scale-free network, and then to
Yeah,
you,
I don't know much about that, but the scale free, which is that I don't know if I use that term, but this is what one means by scale
yeah,
free,
that's
or it's
right.
one of the aspects of it. It's repeatedly found in different networks, like the internet, for instance. And that could be an effect of one hypothesis is that that's an effect of the rich gets richer effect or preferential attachment, which means that when a new node is incorporated into a network, typically it is the nodes that are already highly connected that are going to connect to the new node. So new nodes or new interactions incorporated to the network, they tend to more likely go to proteins that are highly connected already. And then over time, the proteins that are highly connected become more and more highly connected.
Yeah,
So that's what in economy, to do a parallel with economy, we call the rich gets richer effect.
that's right. Okay. All right. If there is no question, then we can finish the lecture today. And then thank you so much for all the information and your lecture. I really appreciate David. Thank you.
Thank you very much for having me, for inviting me.